Thanks everyone for joining us again today. I appreciate you taking the time to be with us here today. And so last week we talked about next gen technologies, and today what we're going to do is take a deep dive into one of the ways that you might use next gen sequencing uh, for public health problems. And so what I thought I would do is invite some colleagues of mine who are at Arbor Biosciences, which is a company that I've been working with for about 15 years. So I have a pretty clear bias that I've been working with them, since the beginning of this technology really becoming commercially available. And so there are other companies that are available. And so what I want you to be able to take away isn't just exactly what Arbor does, but the general approach, right? And again, there are probably some pretty good reasons about why I continue to work with this company while other companies come and go. But the two presenters today are going to be Brian Brunelle and also Megan Beaudry, who's a former student from my lab. And so with that, I'm going to let Brian go ahead and go forward.
All right. Thank you so much, Travis, for inviting us, and thank you for everyone for spending the hour to listen to me talk about hybridization capture. It's really is a useful tool for pathogen surveillance. It's, again, like everything, it's like one of the tools in the toolkit that is could be utilized in the field. So just quick background on myself, if Teams will let me switch slides. There we go. So, as Travis said, I'm Brian Brunelle. My background is in public health. I went to Berkeley, got a degree in infectious disease, studying chlamydia trachomatis. I then did a postdoc at the USDA, studying genetic factors in cattle associated with bovine spongiform encephalopathy. And I spent 10 years working at the Food Safety Enteric Pathogen Unit, studying multi-drug resistant salmonella. You can see there's me with one of our pig patients that we had. And for the past 6 years, I've been working at Arbor Biosciences, and I'm now the senior product manager for myBaits and my tags.
I'll do a quick introduction of myself. So my name is Megan Beaudry. I've been in the environmental and clinical microbiology space for several years now. I did my PhD and postdoc with Travis at the University of Georgia, and then joined the team at Arbor about 6 months ago. Brian and I are coming back from a large conference, and I may have caught some strep and a sinus infection. So Brian's going to be leading the talk today, but I'll be here for questions as we're going to go over some of the topics from my dissertation.
All right, so we'll get into it. So just a brief outline of what we're going to talk about today, just the review of NGS from like what Travis presented, then what are myBaits, why use myBaits, some examples of myBaits being used in practical applications, and then just more information in general about tools that are available. So just a quick background on NGS, which is next generation sequencing. So there are currently in public health various different ways you could detect pathogens from a sample. There are the culture-based methods. This requires a prior knowledge of what you're looking for, developing the specific, media using different chrome agar, and very laborious, but also very cost effective. Then there's the molecular methods, which also require some knowledge of what you're looking for. They're most commonly PCR-based methods and running in gel for detection of presence or absence of certain markers or strains. And then there's the next generation sequencing, which is discovery-based, more expensive than these other methods, but really generate a ton of data that is very useful. Sorry. There we go. Sorry, I think Teams. There we go. There we go. So just a history of genomic sequencing. I won't get into this, but since the 70s, sequencing has been vastly improving as far as technology, creating more and more data as we go along. And this could really be visualized by this graph that shows how many bacteria genomes have been sequenced over time. You can see up until about 2008 or 9, there really weren't that many. And then this is about the advent of NGS, or massive parallel sequencing adaptation into different labs. And now we're into like the tens of thousands of bacterial genomes. And just to, again, go over what Travis presented last week, there are two main types of sequencing currently. There's short read sequencing, This is mostly done with Illumina, but there are now new players in the field, which is great to have more options. Short read is usually about anywhere between 200 and maybe 500 to 600 base pairs of sequencing. So very short, but also very high throughput and very cost effective. There are new players, the third generation sequencers like PacBio and Nanopore and others that might be on the horizon. These can sequence fragments up to like 20 to 50 kb, sometimes longer. So you really can get haplotype and allele resolution, but they're also very expensive. And so there are many ways people use NGS to look at pathogens when they sample. There's Amplicon, there's metagenomic shotgun sequencing, and then there's hybridization capture with tools like myBaits. Just some background on Amplicon sequencing. This is a targeted sequencing approach. You have to use PCR primers. It does analyze genetic variation in very specific regions of interest. It does allow for very efficient variant detection and characterization. However, there's really no flexibility. The primers work or they don't work and take considerable optimization sometimes. And there's a limited number of targets per reaction that you can use. And you can see for this region, the 16S, there you need to have a ton of different primer pairs all working in concert with each other. Then there's metagenomic or shotgun sequencing. This is basically taking a sample and sequencing everything that is in it. This is very good because you get everything that is in the sample and it's unbiased. There's really no changes in the relative abundance of the samples because you're not doing any type of manipulation to the sample. However, this is very expensive, especially if you're trying to sequence something that is very rare. You can imagine if you have a fecal sample that has like 10 to the 12 CFU of bacteria, and you're trying to find something that's 10 to the third, you're really never going to see that in a normal sequencing reaction. And that's why this technology is very good, but very difficult if you're trying to find low abundant traits or rare taxa in a sample. So what are myBaits? myBaits are hybridization probes, and I'll get into that a little bit more in the next slide. But basically, it is just one extra step in the normal WGS or shotgun sequencing protocol. So you still extract DNA from your sample, you make a library preparation, you add in this one extra step that takes about a day and a half, then you go to sequencing. So it could drop right into any current sequencing pipeline. And just as a little schematic, this is how it works. You have a ton of background DNA, which is represented by the gray nucleotides. This is not the correct length, but it's just an example that you have your target of interest, you denature it. myBaits are single-stranded RNA probes that are labeled with biotin. They're complementary to your region of interest. They will come in hybridized to the strand of interest with the biotin. You then add streptavidin-coated magnetic beads. The bead binds to the biotin, which is attached to the RNA, which is attached to your sequence of interest. Use a magnetic bead or a magnet to then sequester the beads that are attached to your region of interest, wash away all the background DNA, clean up the beads, and what you're left is just your target sequence of interest. From there, you then amplify it and go on to sequencing. So instead of sequencing everything that is in your sample, you're really focusing on only those regions of interest, again, whether they are viruses, bacteria, or certain genes. And this is the picture of what the myBaits kit looks like. It's just boxes, buffers, and beads. Basically, you're just binding and washing. What's that? I thought there was a question. Apologies. And so I kind of break down hybridization capture into like two general arenas. One is, you're looking for a portion of a single genome. The human genome is 3 gigabase. If you're looking for only a 3 kb region, which is 0.01% from many samples, there's no reason to sequence the entire genome. You could really just focus your efforts on the subset and really reduce your sequencing by 99.9%. Also, and more applicable to surveillance, is looking for a specific target and a complex sample with DNA from many sources. An example that people have done is like looking at malaria mosquitoes. really difficult to extract blood from mosquitoes, don't really want to culture the malaria. So you could just take the entire mosquito, extract all the DNA in the sample, and then use myBaits to enrich the malaria. Also DNA soil, there's tons of bacteria in soil, and if you're only interested in one particular bacterium or even a subset of genes, it makes more sense to enrich those regions because of their low abundance in the sample. And one advantage of using HybCap is it's good for resequencing as well as variant discovery. So a bait is commonly 80 to 100 nucleotides, and the length of the bait or the probe is independent of the size of the fragment it captures. So it could capture something very small, like 50 nucleotides, it could capture something like 400 nucleotides, and it could also capture something up to about 10 kb. So it's really advantageous that myBaits design can work not only with the commonly used Illumina or short read sequencing, but they're also useful with the current long read sequencers to find longer stretches of DNA from a sample. Also, a very big advantage of the probes or the baits are they can tolerate a high level of dissimilarity in the target molecules. It could be 5% different and still hybridize with equal efficiency as something that is 100% efficient. And this really allows you to be able to hybridize and discover novel variants in a sample. This is just a schematic comparing Amplicon design to a myBaits design or a hybridization capture design. And so this is a virus genome that has different reading frames in different directions and orders. For PCR, you have to create 2 separate primers for each region that overlap in order to get coverage of the entire genome. Whereas myBaits, they're probes that act independently of each other. So we just stagger these. Usually they overlap, or we call tiling, that cover the entire region. And so both of these will work fine. However, if you have novel variation, rearrangements, inversions, or deletions in your genome, many regions of the PCR or amplicon may not work because those regions have been moved or no longer exist or are too variant for correct primer hybridization to occur. Whereas the myBaits, again, because they all work independently, these probes, that you still recover the entire genome. And another aspect of the strength of HybCap versus Amplicon is this is some work that Megan had produced when she was in Travis's lab, and it's 16S comparing Amplicon versus capture versus shotgun versus expectation of a mock community. And the hybridization capture more accurately represents the shotgun than the Amplicon data, meaning there is more bias occurring with the Amplicon and therefore less bias occurring with the myBaits relative to expectations. So having something that is more accurate for the composition abundance is useful. Pros and cons of myBaits. Like everything, there's pros and cons in the right time and for to use any type of tool. So pro of myBaits is it works very well with highly degraded DNA. It can tolerate unknown polymorphisms, so you just have to know about what you're trying to capture. Because it captures the entire sequencing molecule, it recovers flanking loci, which lets you know maybe where an insertion occurred, or if there's a deletion. It's just more powerful in the sense that it's not it gives you information of what is surrounding it versus only what is internal to that particular region. Deep sequencing is not required, which makes the assay less expensive overall. It assists in uncovering the rare members because you don't have to sequence as deeply. Also, decreased data storage and computational time. Sequencing a metagenomic sample to 50 to 100 million reads for every sample really creates a lot of issues with the computational time for analysis and storage if you're doing this repeatedly. Whereas if you're only sequencing, again, maybe 0.01% of a sample, that is much less data to utilize. And the bioinformaticians really appreciate this. You can easily combine any sets of baits. So you could take something from one target and combine with another target, and all you do is just really mix the contents of the tubes. Also specific to Arbor and myBaits is we offer free design and project consultation. The client owns the design. So it's one of those where if you want to work in design, you'll be talking with Megan and myself, scientists who understand this arena, this field, and we'll be excited to help you figure out what you need. It's very easy because it's in a pipeline many people are using. You just add an extra step. It can be used with any NGS platform, and what we provide has all the reagents for the assay. The cons are the kits can be expensive, and it could be, adds extra time to it. But the cost of the kit should be offset by the savings in sequencing or getting data that you would never be able to generate without some sort of enrichment. I'm just going to go through some example publications of people that have used myBaits. This one had detected SARS-CoV-2 in wastewater. This one was interesting that there was a patient that had reinfection with COVID, and they're able to enrich the virus both times. And because they had the sequence data, they're able to determine that it was an entirely separate strain the second time, or variant the second time. So they were reinfected. It wasn't that the original infection came back because it was dormant. This one used a set of herpes virus baits and discovered novel herpes simplex viruses in wild gorillas, bonobos, and chimpanzees. This one had a design for Mpox and was able to identify Mpox in wild chimpanzees, as well as establish the diversity of the virus in the field. This one used myBaits to capture all of the Mycobacterium tuberculosis genome from clinical samples to perform epidemiology and drug resistance surveillance. This one I was a co-author on, which was fun. It was designing baits for Bacillus anthracis because they were looking at samples in Africa from cattle, and instead of trying to culture up anthrax to sequence, they were able to just enrich the entire genome from samples that they extracted from the environment. Much safer, much easier. It could be used in very small samples. This one looked at EBV from plasma in patients, and because of the sensitivity of the assay, they were able to confidently resolve single nucleotide polymorphisms and found that these were associated with a particular type of nasopharyngeal cancer. Detection of low copy viruses from formalin fixed tissue. So another great aspect of HiveCap is you can go to a freezer and find samples that may be 10, 20, even 40 years old, and look to see what bacteria or virulence factors might be present in these samples for retrospective studies. And speaking of bad samples, it also really works on the worst samples you can think of, which are ancient DNA. So this paper was able to recover smallpox virus from hundreds-year-old samples. And this one was able to find HPV from 16th century mummies. So anyways, just some examples of the data. So this paper from Busuli et al. Looked for Vibrio in river water. As you know, pathogenic bacteria can exist at low abundance in the natural environment, but are a reservoir for potential epidemics. Busuli demonstrated that by using myBaits, they found extremely low frequency Vibrio cholera genomes they retrieve from river water. And so this bar graph just depicts that if you were the coverage, if you just use shotgun sequencing for the sample, which would be none, you would never detect that was there. Whereas with enrichment, you can get basically 4x coverage of the genome, which resulted in good analysis of the Vibrio strains that were present. This is a busy graph, but it's really fun that this design was 180,000 baits to work on about 19,000 different viral genomes that are DNA and RNA based, works on viruses that are greater than 72% identity, and it still works on viruses with even lower sequence identity. It just only worked in the conserved region. So it wouldn't give entire viral genome reconstruction, but it did allow identification of certain viruses that were present. And this could be seen by the graph that has no bars. This was not found by shotgun sequencing, but the gray bar or the gray dot indicates that it was found by myBaits, and the light gray bars indicate a virus that wasn't in the design but was still detected by myBaits because of the similarity to the design and this particular virus that they were identifying. And you can also see the gray are the enriched that you could get, thousands of fold enrichment from samples to get really good coverage of the variety of these different viruses that were present in these environmental samples. We have a SARS-CoV-2 panel. This was originally made in response to the pandemic. We basically shut down and did nothing but this for a little while. The design synthesis and QC we did in about 3 days. Standard is about 2 to 3 weeks, but again, we just, put everyone on it and did nothing else but produce this panel. Due to the flexibility of the design, as I was mentioning, that can allow variation. It can accommodate novel polymorphisms, works on all variants, and about 4 years later, the same panel still works on every variant that is in the population. This is different than some of the multiplex amplicon panels, which need to be updated if new variants come out and new primer pairs need to be created. And we also provided the kit for free to the public youth community for about probably a good year, that if people needed our kit in order to sequence, we gave it away. The kit worked very well. The purple reddish bars indicate the coverage of the genome with whole genome sequencing before enrichment, and the green is after enrichment, and there's a very strong correlation. The myBaits maintain the relative abundance of all the different fragments of the SARS in the sample, so it is very normalized. And this just shows virus copies versus coverage and identification of SNPs. You can see even at 1,000 copies of the virus in a sample, the 3 SNPs that were identified could still be resolved. At 100 copies in a sample, 2 of the SNPs could still be resolved, and at 10 copies, 1 of the SNP could still be confidently called in the sample. So it's really powerful for SNP detection of, again, low abundant and rare targets in a sample. So now we could talk about just a few of our pre-designed myBaits kits that are currently available. One is our 16S microbiome panel. This is something that Megan had published on. It's about 40,000 baits. There are baits that cover the entire 1.5 kb region of the 16S rRNA region, and it really allows complete coverage of the RNA gene versus various amplicon panels. So not only is it more comprehensive in identification, the enrichment factor was very large, because on average it was 435-fold enrichment was achieved from a sample. And again, more accurately represents shotguns. Here's samples taken from mouse feces, run with amplicon, capture and shotgun, and the same with from the rat colon. And the capture in the stack bar graph that shows the diversity are more similar to the shotgun than the amplicon is, and vice versa. We also have an antimicrobial resistance panel. It was designed from the CARD database to include basically every resistance gene, as well as pathogenicity islands that were in there. The panel contains over 3,500 unique genes, and it identifies more antimicrobial resistance genes than shotgun alone. So this column basically represents what is expected in the panel. This column represents what, if you just do shotgun sequencing without any type of enrichment, what you will find, which is you're missing a lot. And if you do enrichment, you get really good coverage of almost every gene from this particular sample. And this just highlights that. This is read coverage. Without even looking at the x-axis, you could see clear differences in higher coverage from this darker blue group versus this group that's mostly white. This is the enriched read data, and this is the unenriched read data for those AMR targets. So again, you can get really higher coverage of low abundant targets that you would miss with just shotgun sequencing. And here's a real world example comparing a recovery of number of genes from wastewater treatment plants, from poultry, and from a community. And again, you can see enriched versus unenriched, 300 genes from a poultry sample compared to less than 100 genes from just shotgun sequencing. Really significantly improves recovery of these important loci. We also have a cryptosporidium panel, increase in percent mapping of the enriched and unenriched. So again, just another example of here's the enriched and here's the unenriched, and how from this sample that you would never be able to see the cryptosporidium unless you did some sort of enrichment. And then by enriching, you can see that it is crypto, and you can determine among which different of the six species, these six species that are present in that sample. All right, we have custom reordered and catalog panels. Just real brief on how you can get some myBaits. We have the off-the-shelf one catalog panels. These are usually more cost-effective and can ship within 1 to 2 days because they're always in stock. If you make a custom panel, a reorder takes about 3 to 7 days, so it's still very quick. A new custom panel, newly designed, we will work with you to optimize design for your specific goals and needs. Again, the design and project consultation is free. Design takes 1 to 3 weeks, and it kind of depends on the back and forth. If you have a simple design, you're just looking at several HPV genomes. There's not a lot of back and forth. But if you want to talk about looking at different genes from different species, we might have more discussion how to create the design. The client owns the design. We don't. We don't give it out, but you are free to share it with whoever you want. Travis has always been very good at sharing all his designs so more people have access to the technology. And once we have design, synthesis takes about 2 to 3 weeks. We also have a service lab, and we can allow any, we can facilitate any part of that. If you only want us to do the capture, that's something you do. If you wanted to create your libraries, we could do the capture, then send them to your sequencing provider, or we could do all of it. It's always something that's an option. Some people will work with us on a pilot study and then do the rest themselves. New advancements. So we are coming out in the next month with a respiratory virus panel, which contains 23 modules, and there's 2 options. You could either buy the modules separately, or you could buy a pre-combined all module panel that will cover basically all human respiratory viruses. And just as a point of reference, if you can aplex a capture on the pre-combined module, it would cost about $25 to $45 for the myBaits reaction. You'd still have to pay for the library and all the other aspects along with whole genome sequencing, but this cost, the 25 to 45, should offset any of the sequencing costs that you would need to do in order to get the same type of resolution and identification and of these types of samples. We're also coming out soon with a library preparation kit. As I said, myBaits works with any library preparation kit, so it will work with anything you're currently using. It's more of a convenience for people who just want to order from one place. I work for the USDA, and I really understand how sometimes it's much easier to go through procurement for one order than to go to 2 different places. And it comes with everything you need, though, to create the library preparation for capture. Also, we size it to match the reaction sizes that we sell with myBaits. And how does myBaits support public health? It reduces the need for deep sequencing, so it moves beyond detection and provides much better characterization of what is present. And because of the reduction in sequencing that is required, you can use this on benchtop sequencers. You could do it on a MiSeq. You could even do it on iSeq. If you're doing one sample, it really kind of reduces the need to outsource to large places to wait weeks for information to come back if you have access to a benchtop sequencer. And it does reduce cost. Provides a tool for assessing a variety of public health concerns in many settings. myBaits, as I was saying, is compatible across many sample types, variety of species, and really will work in any environment with any type of sample that you would want. And we can customize any kit to the needs that you have. And with that, I thank you very much for your time, and I'm very happy to answer any questions you may have.